First of all, I'd like to welcome you to this tech talk hosted by the MPCCC. And to begin with, I'd like to pay our respects to the traditional custodians of the lands from which we join together today and acknowledge their elders, past, present, and emerging, as well as any members of the Aboriginal and Torres Strait Islander community who may be among us. So welcome to the tech talk. As you probably know, these talks aim to provide a kind of informal and interactive forum for our research community to discover the latest cancer research technologies and how they can be applied broadly. And today it's my very, very great pleasure to introduce Jiangjing Song from the BDI. So Jiangjing is an associate professor and group leader in both the cancer and also the infection and immunity programs at the BDI. And his research focus is really in tackling biomedical problems at the data level through application of bioinformatics and artificial intelligence and machine learning approaches. And I've had the very great pleasure of collaborating with Jiangjing over the last 6 or 7 years now. And this has led to some really fantastic outputs. And more recently, I've just been really incredibly impressed by the way he's embraced the use of artificial intelligence in the cancer research space. And I've been involved with some collaborations there where he's been using AI approaches to analyze histopath images. And I'm still kind of completely stunned and dumbfounded by the power of AI and how it sees things which are completely beyond us and beyond the pathologists. And so I'm sure we'll be hearing more about that today from Jiangjing. I really would encourage you to try and interact with Jiangjing and apply his approaches to your work, because it really has incredible application and potential, as you'll be hearing about today. So over to you, Jiangjing, and we're looking forward to your talk.
Thank you very much, Roger, for your kind invitation and also for organizing this talk. Today, I'm going to perhaps share with you some of our recent research works that leverage the power of machine learning-based AI approaches in this space in a way that we try to integrate multimodality data to try to address some of the challenging questions. So I guess we are currently experiencing a profound, revolutionary, and paradigm-shifting trend in the modern biomedicine, specifically in the era of post-genomics and also big biomedical data era. So we are facing with the challenges of generating, analyzing, understanding, interpreting, and perhaps also utilizing the high throughput amount of data generated in a very quick and rapid manner. So in this context, my group at Monash BDI is focused on designing, developing, and deploying some of the data-driven computational approaches, algorithms, producing web servers, models, web-based knowledge databases, some of the useful software toolkits to try to address some of the challenging but still open questions in these 4 different research disciplines, including structural biology, genomics, microbiology, and cancer biology. So core to this program is actually relying on the algorithm or software development. Over the last few years, we are increasingly utilizing or leveraging the generated data coupled with some of the machine learning-based techniques. I'm going to showcase 3 different research projects. So these different research projects, there are 2 major types of histopathological image data. The first type is just the so-called whole slide histopathological images, WSIs. And the second major type is getting more and more important nowadays. It's called multiplex immunofluorescence. or multiplex immunohistochemistry imaging. So the second type of image data is more expensive to generate. However, we have been seeing very rapid development and applications of those platform techniques to really delineate or characterize the tumor microenvironment, perhaps in a way coupled with other spatial omics data like single cell transcriptomics and also spatial transcriptomics, or perhaps even spatial metabolomic technique. We're going to see them being applied to this domain more and more in the future. So the first project, I'm going to just illustrate how we can better utilize or combine the power of data-driven machine learning techniques to really, in a way, to extract useful information from the histopathological images. I have been working on this over the last few years in partnership with several key research leaders here at BDI, including Roger and the other group leaders. So we have been supervising a couple of PhD students working in this domain. And we published a number of different data-driven computational approaches, and also one software called HEAL. I'm going to illustrate the utility of the HEAL software package. Just a little bit of background information. I guess you probably already know that still currently the major paradigm is to visualize, inspect, examine the histopathological slides to really, to assess the different stages, types, subtypes of different solid tumors. And in terms of the lung cancer, there are two most important subtypes lung cancer. One is called adenocarcinoma, LUD. The other is called squamous cell carcinoma, LUC. For these two most important subtypes of lung cancer, traditionally, histopathology needs to visually examine the slides and then assess their types or subtypes. However, a couple of years ago, machine learning has been developed to automatically predict or classify those subtypes of lung cancer. There was one perhaps most pronounced finding is published in Nature Medicine. And in that work, the researchers developed the convolutional neural network, which is a canonical or most typical deep learning based technique to train the machine learning model from the lung cancer patients' whole slide images. And then the model was further applied to automatically classify these two subtypes of lung cancer, which achieved an accuracy comparable to the human experts, pathologists. We are actually very intrigued by that work. That work was published in the end of 2018, but now 5 years has been passed. We have been seeing now, I think, foundational model that have been quickly developed and deployed to not just address histopathological imaging now, but also perhaps multi-different modality of cancer-related data. Here, I just want to give you a quick glimpse of the HEAL software package. The full name of HEAL is called Hematology and Eosin Image Analysis Pipeline. So for many of the non-experts in deep learning, it's actually very difficult, very challenging for the non-expert to learn and also further apply deep learning model, even if they already have some in-house datasets available, perhaps in some medical centers or in different labs. So that's why it motivated us to develop the HEAL software package. So we meant to develop HEAL in a flexible and easy to deploy manner. There are only two major labels or input labels to HEAL. The first label is just the whole digitalized whole slide images with some annotation or labels. In machine learning, the machine learning model we need to feed the model with that type of annotation labels. So really depends on the downstream analysis task. For example, this can be a cancer diagnosis or cancer typing, subtyping, or mutation prediction. But nowadays can be risk prediction or treatment response prediction given the label. So it depends on the label. Once we have the label available, then we will be able to train the deep learning model using the HALE software package. And in the HALE software package, there are up to 16 different deep learning architectures or models that users can select to apply to train the model. Then after configuring the model using a local computer, user will be able to train the deep learning model based on the software package. There are several exemplar outputs that can be generated by HALE, including some of the data distribution, conventional receiver operating characteristic curve. So those ROC curves are typically utilized to assess the performance of the deep learning model, machine learning model. And also in that process, users can also be able to visualize, monitor the ACC and loss curve during the model training process, and also some of the confusion metrics. In this way, users can better, understand, the model's capacity in terms of predicting the positive versus the negative, and also some of the heat maps. And also, we also integrate the third-party software called GradCam, because deep learning are known as like a black box machine learning model. It's very difficult for humans to understand what exact type of feature through the multiple layers neuron-based learning process. What's the model really master utilize to make the prediction in order to improve the model transparency or enhance the interpretability or explainability of the deep learning model? We combine the GradCam to really facilitate the understanding of users in terms, okay, from this slide, which region of interest perhaps much more important for the overall classification analysis. So this software can be used to highlight, pinpoint most important regions that are deemed as being contributive or more important relevant for the model to make the final prediction. So those are as part of the effort to enhance the deep learning model interpretation. I would like to highlight 3 different case studies here to help you better understand the utility of the software. So the first case study is a typical multi-class classification task, where we have 2 different types of input data. The first input is just the HE stained whole slide images of the lung cancer patients. We obtained this from the TPG. TPG is one of the most important, perhaps most comprehensive cancer dataset, multi-omics cancer dataset available publicly. The second type label are just for the first case study would be the Luke subtype or normal lung tissue. We just select the ResNet 50 model architecture included in the HALE package to perform the model training. These are just the some of the plots at 2 different levels. So the distribution of the whole slide images at the patient level, as you can see, the data is generally evenly distributed. I mean, in terms of Luke subtype, we have relatively evenly distributed samples compared with the normal tissue images. While the model would need to be trained not at the patient level, but we need to partition or cut the whole slide image into smaller patches or the so-called smaller tiles, because deep learning model would be difficult for the model to, directly input the whole image into the model. This is because what's being digitalized, the size of the image really depends on the resolution, the scanner or the magnification level of the scanning. So one single whole slide image could be like close to or more than 1 gigabyte of size. So it's very large, actually. So traditional deep learning based model, in order to process such a very large digital whole set image, we need to partition the whole image using a sliding window scheme to partition them into smaller patches. So then the deep learning model can be trained at the tile or patch level. And at the tile level, we can see the distribution. Therefore, you can see that for the Luke subtype, there are many more number of samples that can be generated. In this way, the deep learning model can be better enhanced with a much better discrimination capability. These are just typical ROC curves. Again, 2 different levels. The subpanel figure A just shows the tile level performance of the trained deep learning model. As we can see here, for the 3 different subtypes, Luke, the AUC value is actually much higher than 0.85, less than 0.9. However, after aggregating the predictions at the tile level, the model will further enhance the predictive power. At the patient level, the model could achieve an AUC of more than 0.9. That's very good performance, considering that we only utilize the publicly available TCGA patient cohort of lung cancer. So there are only, I guess, less than, I think the data set we utilize is close to 1,000 patients. But you can imagine that in the future, there are many different types of biobank data, like here at Monash. I only recently know that at Monash, there are already very large biobank data available. For prostate cancer, there are like several thousand patients biobank data available. And also for the colorectal cancer, like Professor Helen Abbas' group in collaboration with the Cabrera Hospital, we are currently in the process of handling digitalizing of the colorectal cancer patients of close to 2,000, or in the long term, maybe even more than 3,000 patients, with some labels regarding some cancer reoccurrence or relapse, or even some treatment responses. So that's very rich and very valuable data set for future study in terms of developing some of those data-driven AI approaches to really analyze and better understand some of the very important biomarkers in terms of patient treatment or the relapse, this kind of analysis. The second key study of Hill packaging is a multi-label, multi-class classification. So in this task, we try to better understand whether or not we could train a deep learning model directly from the histopathological images to predict some of the relatively well-known, perhaps very frequent mutated oncogenes. Again, we utilize the dataset downloaded from the TCGA public database. And we can see here, there are a number of very well-known oncogenes, including the TP53 and a few other kinase genes. These genes are being considered as very frequently mutated. Then we train the deep learning model. And from this ROC curve, we can see that out of the 10 most frequently mutated oncogenes, we can see that at least 6 or 7 oncogenes, their mutation could be directly predicted from the histopathological image data without the need to sequencing their genome, the panel test, gene panel test. So that's really shows that it's possible to develop the computational approaches directly from the histopathological images to identify the corresponding oncogene mutation given a very well annotated labeled data set. Again, I also want to highlight that for some of the oncogene, we can see that like the STK11, the AUC value is actually very high. So that's pretty accurate. Although a number of other ones, the AUC performance is pretty marginal. However, again, I want to remind that we only utilize a relatively limited data set from the public TCGA database. And if in the future we could have a much larger patient cohort with this oncogene mutation, with annotation of sequence data, coupled with the histopathological imaging, we would be able to deliver or perhaps train a much better and more precise deep learning model. And the third case study, we then further examine whether or not we could predict other different subtypes of like the colorectal cancer. Particularly for the colorectal cancer, there are two different molecular subtypes. One is called microsatellite instability, MSI, or microsatellite stability. These two subtypes is very challenging, nearly impossible for the pathologist to use their own eye to directly tell, discriminate from the histopathological images. However, after we train the deep learning model, as you can see from these ROC curves, for these two subtypes of colorectal cancer, the AUC value could be achieved in the range between nearly 0.65. That's still, we only train the model using a very modestly sized data set, incorporating, I think, less than 500 patients' whole slide images. Again, with the increasing availability of more annotated whole slide image data in the future, the model accuracy, we could expect it can be further improved. The second research project is focused on the gastric cancer. Again, from the histopathological images. And for gastric cancer, we know from the molecular characterization perspective, there are four major molecular subtypes of gastric cancer, including the EBV virus-associated subtype and microsatellite instability, MSI, chromosome instability subtype, CIN, and genomic stability subtype, GS. That's from the molecular characterization perspective. It's very costly to characterize these molecular subtypes. So therefore, we wonder if we would be able to develop a deep learning-based AI approach directly from the histopathological images to tell or predict these four major molecular subtypes of gastric cancer. That's why we developed this software, this algorithm. This is just the general workflow of the overall AI approach development. As we can see here, there are five different major stages towards the development of this AI approach. Again, the first stage is just the dataset curation. We have very limited access to the in-house separate patient cohort. So for that work, we again use the currently available gastric cancer cohort data from the TCGE public database. And we can see that there are only less than 300 patients with this annotation as four molecular subtypes. Then the second stage is just data pre-processing. And after we remove some of the ambiguous or blurry tiles, we return the final relatively high quality data set for deep learning based model construction. And in order to assess the model performance, we have to generally split the whole benchmark data set into three different subsets, including the training, the validation, and the test. So during the model construction stage, we tried multiple different deep learning architecture. And in the end, because the data set was still very limited, that really tied the hand off. But then we tried many possible alternative deep learning model architecture. And finally, we select the efficient net, this architecture as the final model architecture to train the deep learning model. For each of the four molecular types, there was one model trained. In the end, we would be able to further apply the optimized model and then assess the model performance by performing separate independent tests using this dataset. This is the overall performance of the model. Again, because the dataset is very limited, so in order to avoid the overfitting risk, overfitting issue, so that is typical for many of the machine learning-based practice. So especially for very limited data set, so the deep learning model will be relatively inclined to be overfitted. Therefore, in order to enhance the model robustness or generalization power, we introduced ensemble learning-based deep learning approach. So this ensemble learning approach are beneficial because it can be useful for, on one hand, enhance the model performance based on the relatively limited benchmark dataset, but on the other hand, it significantly reduces the possibility of having the overfitting issue. And we can see here from this performance table, without the ensemble training strategy and compare with the ensemble training and without ensemble training, generally the model performance could be further enhanced. And on the other hand, after we further apply the trained model to the independent test dataset, we observed that the performance drop was actually minor compared without the case of without the ensemble training. Therefore, this ensemble learning strategy can be very useful for enhance the model performance and reduce the overfitting issue. These are the performance evaluation using the test dataset. And we can see here for the 4 different molecular subtypes of gastric cancer, the model, the deep learning model, for the majority of these molecular types, the model would be able to identify them with a reasonable AUC value. Depends on molecular subtype, as we can see here. At the patient level, the AUC value ranges between 0.7 to nearly 0.9. So these results altogether indicate that it is possible to train a deep learning model to predict the molecular subtype of gastric cancer directly from histopathological images. After the deep learning model is constructed, we could easily apply the model to perform the digital staging, digital scanning in an automated manner. This is just the exemplar figures that are generated by the deep learning model in terms of molecular subtype analysis for these 4 molecular subtypes of gastric cancer. The third research project, we move on to a different image data type called multiplex immunohistochemistry images, multiplex MHC images. We know that from an intuitive perspective, the patient's tumor microenvironment, generally there are some hidden information in the tumor microenvironment of the patient that should be reflective or indicative of the patient's the individual patient's status. So that motivated us to develop develop a different type of machine learning based approach called graph neural network based approach. We term this approach cell graph state. I'm going to illustrate how we can construct this approach. We further compare the performance, the patient staging performance of our approach compared with the conventional TM approach. For the TM approach, it's still currently adopted. The T means tumor, A means node, M means metastasis. So the TM staging approach is still currently predominantly adopted by many hospitals and medical centers all over the world. And we compare the performance of AI approach-based patient stratification and with this TM stage. And we show that, especially for the triple class patient survival stratification prediction, our method perform better in a statistically significant better than the TM stage approach. I think that's something very important. Again, for this work, we are still we only have very limited access to the in-house patient data. This work was in collaboration with Shanghai Ruijin Hospital, affiliated with School of Medicine, Shanghai Jiangjing University, in collaboration with Professor Daquan Xu's group. Professor Xu previously worked at the Hudson Institute before. So we have been collaborating on this project for the last 3 years now. This is just the basic demographic information about the patient cohort collected from Shanghai Ruijin Hospital. Particularly, I would like to point that in order to assess the AI approach, the patient stratification performance, we perform two different type of analysis. One is predicting the binary class patient survival prediction. So the binary means, patient depending on the survival time, separated as classified as with survival less than 2 years or survival longer than 4 years. That's the binary class. While we also compare the performance of our approach with TM staging based on the triple class survival. Triple class means, patient short-term survival less than 1 year, intermediate survival between 1 year and 5 years, longer survival over 5 years. This is the overall workflow of our developed approach called cell graph stage or cell graph. Again, there are multiple different stages for the development of this AI approach. The first stage is just the specimen processing, like collected tissue, capture patient tissue, and followed by MIP staining. Then we digitalize them. Note that here we would be able to only apply 7 different protein biomarker to stain the patient tumor microenvironment. My understanding is that nowadays there are some commercial toolkits that could stain many more biomarkers, not just these 7. So that would be much more important in terms of the providing much better, richer information about the tumor microenvironment. But anyway, this study we only utilize 7 biomarkers. And then after that, we pre-process the images and represent each of the cell. There are many different types of cells in the tumor microenvironment. We represent them according to their markers and represent them as cell communication or cell interaction graph. Then after that, we would be able to model their interaction pattern, their cell communication network using graph-based deep learning cutting-edge AI approach. So because again, I want to mention that because the cases there are only in the end we could only utilize less than 200 patients. Anyway, we performed further participating the whole data set into training, validation, test, and evaluate the performance at the independent test level. There are 4 different graph-based neural network model architecture, including GCN-SAG, GCN-TOPK, GRN-SAG, GRN-TOPK, and we performed some preliminary benchmarking analysis, and the result shows that this GRN-TOPK can, among the other alternative model architecture, can lead to overall best performance. Then in the end, we finally select this GRN-TOPK as the model architecture to train our graph-based deep learning model. This is the overall architecture of deep learning model. So these are the performance evaluation. Again, these are ROC curve for the binary class patient survival prediction. As we can see here, the AUC value is very high, right? So it's close to 0.96. That indicates that the deep learning model trained using those molecular biomarker staining MIC images, we would be able to develop very accurate model for predicting the patient survival in terms of 2-class classification. These are the different performance comparison of different model architectures. This is our further performance evaluation using the separated independent test cohort. Again, the performance drop on the independent test cohorts, there was a very minor, very little performance drop. That indicated the model was pretty robust in terms of predicting the 2-class patient survival. AUC value was slightly lower than 0.96. Again, for the short-term survival and long-term survival, the model is pretty accurate. And I mentioned that we also trained a triple-class patient survival prediction model. Here, I want to show the results, performance comparison between our AI-based survival prediction method with conventional, the mainstream TM staging approach. These are the Maya-Kaplan survival curve. As we can see here, for the TM method, there are 2 subtype methods, one is TM2 or TM6. And we can see that there are 2 primary index here, C index and also the confidence index value. And our approach, cell graph 2, although the performance was better than the TM2 approach, but slightly worse than the TM6 approach for the two binary class survival prediction. However, further integrating or combining the prediction results of our approach with the TM2, we could further significantly enhance the patient stratification performance, with the C-index being further improved to nearly 0.75. With a significant p-value of less than 0.0001. That means that there are some complementarity here offered or generated by the AI approach trained using the cell-cell communication graph-based deep learning model, and with the traditional TM approach. These are the triple class patient stratification performance. Again, as we can see here, for the triple class patient stratification, survival stratification, short-term survival, intermediate survival, and also long-term survival, the cell graph, our AI-based cell graph approach achieved a much higher, much more significant value, much higher than any of the TM sub approaches, like the TM3 or TM6. Again, with a very significant p-value. So this highlighted that indeed there are some important information that could be potentially extracted or represented by AI-based approaches by modeling the tumor microenvironment based on the different molecular biomarker staining MI3 images. I guess that's the major methodological hints I would like to share. And we are currently in the process of doing some paired analysis. Again, in collaboration with Professor Xu's lab, we are generating a patient cohort data of over 200 patients with paired image data. Here, by paired image data, I mean we have now generated both histopathological images and also the multiplex immunofluorescence, immunohistochemistry image data. Our hypothesis here is that by modeling the tumor microenvironment using coupled paired, like two modality of image data, we would be able to further significantly enhance the patient stratification performance. We are doing this work. Currently, we are working on developing a more enhanced AI approach. We will see how it performs. I think that's all I want to share today. Finally, I would like to thank my collaborator, including Roger, Jeff, Chris, Terry, and also my PhD student, Jianning and Zhikang. Also my collaborator from the Alfred Hospital, including Alex and John, also Professor Dakang Xu from Shanghai Jiaojing Uni, and also my collaborator from the University of Tokyo, Human Genome Center, Professor Imoto, and from the Cambridge AI Center for Biomedicine, Professor Petro Liu. Also collaborator from New York University School of Medicine, Professor Srigos and Nicholas. Yeah, thank you very much.
Great, thank you very much, Jiangjing. An amazing presentation as always. I'm always stunned when I see those data sets. I think it was the first time I saw the data from the lung cancer patients where you could predict with a reasonable confidence which mutations, what mutations were present in the cancers. It's like, particularly with the STK11, it's like, oh my word. That so if anyone's got any questions, please raise your hand or put them in the chat. I just want to start off with a question which really just stems from the observation about being able to predict the mutations present and so on. So where are we in terms of actually being able to identify the features then which the AI is actually spotting? Because obviously there must be something to do with the cell morphology, and whatever that the program is detecting, which we can't, as human beings, we can't see. So where are we in terms of actually being able to identify what the features are? Because obviously that's then going to open up a whole new area of kind of discovery research and whatever to characterize that further. So do you want to comment on that?
Yeah, thank you very much. This is a very insightful question. I didn't provide the feature list, but like you mentioned, most of the features that are deemed as being contributive to the performance to the diagnostic prediction performance of the AI model related to cell morphological feature. There are also other type of features. I think that's related. We separate the feature really based on the molecular marker. For each of the antibodies biomarker, there are some features that are descriptors of the size of the nucleus, cytoplasm, nucleus intensity, cytoplasm intensity. Yeah, I think not just these individual features, but more or less it's also like the organization, right? Like the network, the cell, the spatial, the geospatial location and the organization of those cells relative to the other neighboring cells in terms of different subcellular subtype of cells, and immune cell, many other cells. I think really also the organization or the architecture of the spatial arrangement. I think this is something that's why based on the graph-based neural network, it can better model those relatively complex interplay, try to capture the underlying pattern.
Sure.
Yeah.
Thanks, Jiangjing. Mitch, you've got a question.
Yeah, thank you, Roger.
Yeah, thanks for a really interesting talk. I guess this is probably related to what you were just saying about the architecture and the patterns of the tumor cells forming in the tissue. So I think most of those examples were for primary tumors. And I was wondering whether the machine learning and AI at the moment how it performs if, for example, you had a tumor at different metastatic sites. So they're growing in very different niches with different microenvironments. For example, I work on prostate cancer, and the pathologist might be asked to look at a lymph node or a liver metastasis or, whatever, to look at the pathology. And how would the algorithms perform in those very different contexts? Or do you need to have lots of examples of tumors from those different contexts to train the algorithms?
Yeah, I think both aspects of information would be useful. Also, many of the work currently, they didn't model the development of a TME in a time series, in a dynamic manner. In the future, of course, these are very expensive to generate such a high quality time series data set. But I resume that using the PDX models, if our cancer organ noise, if we can reduce the cost and the sample the growth of the tumors in different across different time points, and then using machine learning model to model them, followed by treatment, prior to the treatment, this type, we would be able to answer very fundamental question. So I presume in the future there may be some research efforts applying that direction. But how costly it would be, I don't know. But it would be very expensive, I guess. Yeah, to generate such a data set. Yeah. But that's certainly very interesting to explore. Yeah.
Jiangjing, I've been very impressed about the work you've been doing in the tumor microenvironment, and this is obviously a massive area for research at the moment. And as you highlighted, people are increasingly using these multiplex panels to characterize the TME, which obviously brings with it its own challenges. I mean, human beings can't really take on board or analyze the multiple kind of combinations of markers. We're okay as seeing something going up or something going down, but when you've got, like, I don't know, 6 or 12 or 20 markers and they're going in different directions, you really need the AI and machine learning approaches to analyze those data. So one area that I'm sure would really benefit here is identification of predictive biomarkers for response to immunotherapy. And I was wondering whether you were planning to work in that space or whether you were already collaborating with people in that space, because, beyond kind of classical things like PD-1 or PD-L1 and so on, we haven't really developed that in the cancer space, and it's a really critical question. So I was wondering whether you were working there.
Yeah, I had a discussion with Renee a couple of weeks ago. I just realized that in Renee groups there are more than 300 different PTX models with those histopathological images being digitalized. So that's something we can work together, I think, to model the cancer recurrence potential, key genes underlying those pathways that really underpin the development of such resistance. I think that may be possible, Roger. Again, I think Monash has very valuable partnerships across different medical center hospital. In the long term, would be very encouraging that we team up together to really, develop the fundamental research program by integrating the resources all together, all the talents together.
I think so. I think you're absolutely right. I mean, as you mentioned at the beginning of the talk, we have got incredible resources in terms of biobanks and TMAs and so on.
Yeah.
And it really does highlight incredible potential to go back and reanalyze those and then apply your approaches. And
yeah.
If we do have any clinician scientists on the call here, on the Zoom call, I'd certainly encourage you to think about that and to contact Jiangjing. He's got a kind of incredible team of PhD students which he maintains. I don't know where Jiangjing finds his PhD students from, but he's always got this incredible team of incredibly dedicated and brilliant PhD students who work on these projects. And so you can get his team onto your questions straight away. How do you, just like kind of a bit of a left field question. I remember seeing a presentation on AI once where they highlighted where it could go wrong, and they were actually using AI to basically distinguish between dogs and huskies. And the program was going really, really well until they worked out that actually what the program was actually spotting was the snow in the background of the huskies and not actually the huskies themselves. So there's clearly kind of, because we don't know what these programs are spotting, there's clearly avenues for misinterpretation and it going awry. So do you want to comment on that, and what kind of controls do you have in place to avoid that?
Yeah, thank you very much, Roger. This is very insightful question, very important. Those works I just showcased today, they are actually done in a supervised machine learning manner. Being supervised, I mean we have to manually feed the label into the model, supervise the training of the model in a human interventional way. However, the cutting edge AI nowadays, there is no human interference. These are called self-supervised learning, specifically applied to the foundational model. For example, in the tumor microenvironment analysis, Harvard Medical School, they are a very leading group internationally. They already use their in-house, Harvard Medical School, they launched like AI Center, right, a couple of years ago in collaboration with MIT. Their in-house pan-cancer image data are more than 100,000 images. And they train the foundational model in a self-supervised manner. You don't intervene, humans don't intervene with the model training. So then these are very robust. Then they further apply the generalized model. First stage, training the self-supervised generalized model. Second stage is to address very specific downstream clinically relevant questions. And they show that, for up to more than 30 different clinical relevant questions, including relapse risk, whatever, show that the generalized self-trained machine learning perform much better than the supervised model. I think that's of course the frontier of this field. But again, like another going effort is that we actually realize standardization. There are some formatting issues. There are some general standardization. So a lot of confounding factors can indeed influence the performance of the model. Data collected by different hospitals, by different experts, digitalized. That's those confounding factors. We have to address that. In order to address that,
it always worries me about how people label the slides and things like that.
Yeah, exactly, Roger. Yeah, that's why I think there are still some issues. Yeah.
Okay, sorry, Mitch, have you got another question there?
Yeah, it's a kind of a similar, practical question. I was wondering about the I guess this approach relies on the imaging as well of the original slides, and I was wondering, you know, what resolution imaging that you use, and because I guess at least for prostate pathology, when it's done by a human, is the idea was that it could be done at a low power, and so you're looking at features that I guess would require better resolution and more granularity. Is that right?
Yeah, so about the resolution, I mean, the typical manufacturing level is 40 plex, right? 40. So
40x, yeah.
20 now, but now you can train the machine learning model across different resolution levels. They integrate together. So that's one way. Another way is the finer the resolution is the better. I mean, generally, yeah, would be. Yeah.
Okay, thank you very much. Well, if there's no other questions, I'll draw this session to a close. I just want to thank Jiangjing for his presentation. It's been very stimulating as always. Thanks everyone for their attendance. And please visit the MPCCC website to check for more upcoming events, and in particular, check out the new MPCCC website, which Hannah's very, very proud of, and she wants us all to go there and have a look at it. So thanks a lot for